students, a warm welcome to all of you. I am Dr. Pooja from Department of Zoology, Hansraj College. Today I will tell you about Mendelian genetics. Genetic characteristic. We all know about genetic traits. These are the traits that we inherit from our parents. They pass on from one generation to another. Yes, it can be physical traits like our eye color, our hair texture, our skin color. These all are genetic traits. These genetic traits, it can be biochemical in nature, it can be physiological in nature. These are the traits that we inherit from our parents. And yes, these pass on from one generation to another. This is also known as biological inheritance or heredity. Now, this act of inheritance was seen by the people in past. But what could be the factors? What could be the pattern behind the inheritance of trait? That was a big question in the past. Many people tried to find out the answer for the same. It was George Mendel who find out the patterns or the logic behind the transmission of these traits. Mendel, he was born in 1822. He was excellent student in high school. He studied philosophy, botany and physics. He taught physics and natural science for over 15 years. And actually, when he also thought of the same thing, what could be the patterns or the logic behind the transmission of trait, he carried out his first set of experiment in 1856. So, the problem was, what are the factors or the patterns responsible for the transmission of trait? Now, before answer, he first planned to answer those questions. So, to execute his work, first he made an experimental plan. So, he chose right model organism, and in his case, he pointed on garden pea. That was a model organism he chose, Pisum sativum. Why he chose garden pea? Because it was easy to grow. It reproduces well and grows to maturity in the single season. Self-fertilizing in nature, but yes, it is easy to crossbreed experimentally. Large number of progenies can be obtained because he want to study different generation for a particular trait. So this was very important point. In this case, he can collect large number of progenies. And one of the very important thing in garden pea, he could find out the visible genetic features which are represented by contrasting traits. And yes, the choosing a right model organism is an important aspect of experimental methodology. For example, in garden pea, as I mentioned, he focused on 7 physical traits, and these were seed form and seed color. So seed form, as I mentioned, there was contrasting traits, so it could be round or wrinkle. Seed color, yellow or green. Flower color, white, purple. Pod form, full constricted. Color, yellow, green. Stem, position of flower, it may be axial or terminal. Size of stem, it could be long or short. These were the 7 genetic visible features that Mendel chose to focus. But again, in his execution, when he worked on execution, he didn't focus on all the characters at a time. Actually, he focused on one character at a time. That was the beauty of his experiment. So, and then he started his experiment focusing on one character. He bought seeds from true breeding strain where a particular strain will remain same year after year, generation after generation. That's another plus point from his experiment. Choosing one character at a time. Secondly, he brought the pure lines. He bought the seeds which were pure lines where a particular trait will remain same year after year. And he focused on one character at a time for examination. And the third very important point in his experiment, he kept quantitative record of each genetic experiment. And that was his first set of experiment, and that is known as monohybrid cross, because he started with the parents which were same for all the characteristics except for one genetic characteristic for which the parents were showing contrasting trait. For example, for height, that is a one genetic characteristic, and the contrasting trait were tall versus dwarf. And that kind of a cross that is made by crossing two breeding strain that shows alternating trait for a genetic characteristic, that kind of cross is known as monohybrid cross. So to start with, he started with parents that shows a particular genetic characteristic, but that was tall, all were tall, and they were like a true breeding strain for the characteristic tallness, and they were crossed with another set of plants that were short for that particular genetic characteristic. So he started with this parent population, and in F1, that is the first filial generation or the first progeny population, all the plants he observed, they were tall. That was the observation. In F1, in first generation, all the plants were tall. When he allowed these F1 plants to undergo self-fertilization, he found that that will be the second generation, and that is also known as second filial generation. He found that in the second generation, progenies, he obtained so many progenies. Here only 4 has been represented, but he obtained so many progeny, and he found that most of them were tall while few of them were short. Now, being mathematician, being having knowledge of statistics, maths, he actually not only qualitatively observed the result, he made the quantitative record of this information as well. Means how many were tall and how many were short. And then the observation was, number 1, F1 plants were consist only of tall plants. Number 2, in F2, most of the plants were tall, but yes, the dwarf trait got reappear in F2 and it was not there in F1. And as I mentioned, he made quantitative record of that data as well. So he obtained around 1064 F2 plants, and out of which 787 were tall and 277 were dwarf. And he made the ratio of it, and he obtained approximately 3:1 ratio. 3/4 of the plants were taller while 1/4 of the plants were smaller. And obviously he repeated this experiment, and he got the similar observation on repeating those experiments as well. Then that was for the one characteristic. He also made similar crosses with other characteristics, rest of the 6 characteristics, and interestingly he got similar outcome in F1. Means the similar outcome means one of the contrasting trait appear in F1 while both the traits appear in F2, but in the ratio of 3:1. So that was the observation table. You can see for F1 all the plants were showing one trait, and in F2 both the traits appear for that particular genetic characteristic, but in the ratio of approximately 3:1. Actually he could see some pattern that, oh, I am getting similar ratio for all the characteristic. There is some pattern that he could see in the observation. Next, he also did reciprocal cross. Reciprocal cross means the cross where a female expressing a particular trait is crossed with a male not expressing the trait, followed by a cross where male expressing a particular trait is crossed with a female not expressing the trait. For example, if he started with a plant that was initially tall, and if they are contributing the male gamete, in the next cross, he took the plant which was tall but that is contributing by female gamete. Such crosses are known as reciprocal cross. Such crosses help us to understand the role of sex in the inheritance of genetic traits. And he found the similar result in reciprocal cross as well. Mendel results were not sex dependent. That was the next observation. Now, this is not the end. That is just the observation. And when we do some experiment, yes, we plan it, we execute it, we observe the result, and then we try to explain that result. And when he tried to explain the observation point, he made hypothesis. He gave the following hypothesis to explain that observation. He said there are some factors responsible for a trait, and there are some factors responsible for a trait, and there are 2 factors for each characteristic. For example, for height, there are 2 factors which are responsible for it. And in true breeding population, which is started with, both the factors are same. So for example, tall plants, say these factors are represented by capital T, tall plants they were capital T, and for dwarf plant they may be small t. Second hypothesis is, these factors segregate at the time of gamete formation, so that each gamete carries single factor for that characteristic. And third hypothesis, or the point he tried to explain, is when 2 unlike factors are present in a single individual, when 2 unlike factors they come together in a single individual, only one factor expresses its trait, that factor is known as dominant factor, while other gets masked, that is known as recessive factor. So he made these 3 points and tried to fit in the observation. Now we will try to fit this in the observation that we observe. So to starting with, he said there are 2 factors for each character, and true breeding lines or the pure lines, they carry the same factor for that particular trait. For example, here the tall plant is a pure breeding line. So both the factors are same. Both the factors are same, capital T, and for short, both the factors are same, small t. Second point, these factors segregate at the time of gamete formation. So we can see that during gamete formation, these factors segregate so that the gamete from these parent, they carry only capital T, while the gamete from this parent, they carry the small t, one factor for that particular character. These gametes, they fuse together at the time of fertilization, and there is a formation of next progeny, and we can all see all will be capital T, small t, capital T, small t. Third point he made, when 2 unlike factors were present, only one of the factor express itself, that is known as dominant factor, while the other remain masked, that is known as recessive factor. So in F1 we can see there were 2 unlike factor, so only one will show its expression, and in this case all plants were tall. So only capital T has marked its expression. So that capital T is known as dominant factor, while the small t is known as recessive factor. And on doing self-fertilization between different F1, again there will be the gamete formation, there will be segregation of these factors. So the gamete from these parent, they will be carrying either capital T or small t for that height, for that characteristic. And similarly the gamete from this individual will be carrying capital T, small t. So on fusion, we can see when the gamete of this parent, it fused with the gamete carrying capital T, there is a formation of capital T. If there is a fusion between capital T, small t, there is a formation of capital T, small t. And if there is a fusion between this gamete and this gamete, again we get capital T, small t. And if there is a fusion between this, we get small t. So, and this happen, and there are three times more probability that the plants will be either tall, because they will either be capital T or capital T, small t. And yes, 1/4 of the times the plants were dwarf. So these 3 points, they came under the postulate that could explain the result of monohybrid cross. That was, there are 2 factors for each character. So basically the first postulate he gave after monohybrid crosses, there are unit factors in pairs for each characteristic. And yes, out of these 2 factors, one factor act in a dominant manner, that is known as dominant, while the other is known as recessive. So that postulate come under dominant recessiveness. And the third point, very important point he gave, that these factors segregate at the time of gamete formation. So basically from his initial set of experiment, he could explain how there are factors which are responsible for the inheritance of trait. And for genetic characteristic, there are 2 factors in us that is responsible for a characteristic, and these factors may be same, may be different. At the time of gamete formation, these factors segregate so that each gamete carry single factor for the character. So that was his initial observation after monohybrid cross. And yes, nowadays we know that these factors are nothing but these unit factors are nothing but genes. And yes, there are two forms of gene, there are two alternate form of genes, they are known as alleles. And when the physical expression, when these factors get expressed, they give rise to some physical expression of a trait that becomes phenotype, and what combination they are present in individual, that constitute their genotype. And when both the factors are same, the individual is said to be homozygous for that particular characteristic, and if both the factors are different, the organism is said to be heterozygous for that trait. So dear student, we have learned how executing a simple experiment but in a planned manner, he could find out the important answer of a long-term question, what is responsible for heredity. And from monohybrid cross, he could find out that yes, there are some factors nowadays which are known as genes. These are responsible for heredity. Thank you. Have a good day.